You're good. just okay, I enabled the chat and the Q&A. So
the poll probably.
The poll also is visible.
Wonderful.
So I have to invite Andrew, Dr. Lee here. Andrew.
Hey, Chris. Chris from University. Hello, everyone.
Yeah. So we're just waiting. Hello, Dr. Lee. How are you?
Good. How are you guys? Can you guys hear me?
Hey, yes. So I think everything is fine. Sound and the video, and we are live, and I think people are joining us. Let me see. Yeah, we have people coming. So I think we give some minutes for some housekeeping notes, and then we start our seminar today. So hello everyone. Today it's a pleasure to have Dr. Lee with us to talk about fresh bioprinting. Before that, I will actually take some time for some housekeeping and announcement, and then we move forward for his presentation. So let me just share my screen. Okay. So can you see my screen now?
Yep.
Okay, great. So as always, you could ask your question in the Q&A box, and we have a poll box here also. You could also take the poll, and it really helps us to improve our e-seminar series. And also we record these e-seminars and we put in our YouTube channel. Make sure to subscribe to our channel, and then if you need a refresher or if you miss the session, you can always watch them on YouTube channel. And our next speaker is Dr. Massimiliano Paganelli. He's a clinical scientist. He's going to talk about a stem cell-derived encapsulated liver tissue, preclinical development, and allogenic cell therapy to treat liver failure. And he's the founder and CEO of Morphocell Technologies. And next week, same time, we will host Dr. Paganelli here. As always, you can follow us on Twitter and social media, and if you have any question, you can ask your question from the organizer, Dr. Akbari, myself, and Mr. Kamzadeh. And we would like to also thank our sponsors, Montreal Transmedtech Institute. The Montreal Transmedtech Institute aims to support the development of innovative medical technologies, train the next generation of professionals, and make innovation in life sciences and engineering. Based on a living lab approach, Montreal Transmedtech provides an integrated environment that supports interdisciplinary collaborative processes and co-creation of new medical technologies and interventions to catalyze their development and adoption by users. We also thank University of Victoria, the Innovation Hub, that sponsored this talk today. So by that, I would like to introduce our speaker. Let me just unshare my screen. So you don't see my screen right now, right? Okay. So today, it's my great pleasure, actually, to have Dr. Andrew Lee with us. Dr. Lee is currently the head of bioprinting and co-founder at FluidForm, a 3D bioprinting company looking to change the way we think about tissue engineering. He received his PhD in biomedical engineering at Carnegie Mellon University in the regenerative biomaterials group led by Dr. Adam Feinberg. His PhD thesis work was focused on muscle tissue engineering, leveraging bioprinting to fabricate skeletal and cardiac tissue models with complex tissues, architecture, as well as physiologically relevant contractile functions. Part of his thesis was on 3D bioprinting contractile tissue and computing heart, was recently published in the journal Science. More broadly, Dr. Lee is interested in the intersection between biomaterials, cellular biomechanics, computational design, and tissue development for the creation of the functional cellular and tissue systems with composition and architecture that resemble those of native tissues. Beyond research interests, Dr. Lee wants to see the growth of bioprinting as an easily accessible technology that has scientific impact for both basic and translational research. So by that, I would like to invite Dr. Lee to share his screen. But before that, I was working on the 3D bioprinting in my postdoc. I have a story to tell. So, and I was looking to bioprint an elastomer, which was very difficult to bioprint. So I found the work of Dr. Feinberg in 2017, and then I used the technique and it worked for me. And later on, the seminal work of Dr. Lee came up in Science. So I was excited to see the technology is great and working, and I used it. So I have a hands-on experience on fresh bioprinting. So Dr. Lee, please go ahead, and the floor is yours.
Thank you so much, Human, for that really, really kind introduction. And thank you for organizing this, and with Dr. Akbari for organizing this and putting on a platform for us to kind of share science during these times. I think it's awesome to be able to jump on and be able to watch a talk from all these great professors and talking about their research and what they're doing to take research out into a translational direction. So if I share my screen.
Yeah.
Okay. Yeah, so today I will be, like Human said, I will be talking about fresh 3D bioprinting. And this is a technology that was developed at Carnegie Mellon University and that we've taken out to FluidForm. And so really, I think what I want to talk to you about is thinking about bioprinting through a different lens than what I think our field has thought about it for a while now. And so I think hopefully in these next 30 minutes, I can talk to you about a different way to think about bioprinting research, why we think that is important, why we think without this kind of viewpoints, we might not be able to grow as a field as rapidly as we want to, and why this shift in mindset might be able to take us towards clinically relevant research. And so before that, Human gave a really nice introduction, and so I'll kind of very briefly go over my journey, where I started from. Currently, I'm bioprinting lead, head of bioprinting, and co-founder at FluidForm. And where I started was back in undergraduate, where I attended UC San Diego and received a degree in bioengineering, biotechnology specifically. And so at the time, I was fairly young. I didn't really know what I wanted to do. I had the privilege, the opportunity to receive a scholarship that allowed me to start to join a lab very early on in my freshman year. So I joined the lab of Professor Adam Engler, who is really one of the leaders in the field of stem cell biology and mechanical transduction. So looking at how stem cells interact with their environment. And what was really cool was the research itself, and then also what kept me going for 4 years as I continued to do independent research for 4 years in the lab was I found that the lab environment to be a really, really really collaborative and friendly one that I really enjoyed. I was able to work with a postdoc who was mentoring me and just kind of learn the scientific process, learn how to do research, and really enjoyed working with people through that process. And that's kind of what led me to decide to continue to pursue a degree at Carnegie Mellon University. You heard Hooman talk about my doctoral thesis, which is on the reprinting of collagen to engineer muscle tissues, and specifically looking at some of these complex architectures of muscle tissues, so skeletal muscles and cardiac muscles, and then thinking about ways to leverage advanced manufacturing technologies to engineer those architectures. And then so I had the privilege of working on a project that led to publication in Science, which I will talk about significantly, since this is really the core technology that FluidForm as a company is founded upon. And I think that the paper itself really conveys our vision of propelling bioprinting research towards becoming very key to regenerative medicine, tissue engineering, and really biomedical research as a whole. So after I finished my PhD, I graduated from my PhD program about a year ago now, and I decided to join FluidForm and co-founded FluidForm because we really felt that we had a platform technology. So I think if you've attended previous talks before, you've heard Dr. Ali Kadamiseni and also Professor Jeff Karp last week talk about having a platform technology as well as a kind of a high impact publication that really backs up your technology. And then that really is two of the key requirements to bring a technology out into a startup environment, and then be able to find a place for it in the startup environment. And so since I just recently graduated, I find it that the best way I can kind of convey my experience is just I have just talk about what my experience in graduate school and some of the take home points that I have. So I think for some of the younger students, and of course I am just recently starting my career, and these are my personal experiences, and hopefully we can kind of share them. So some of my personal experiences that I found it that it's okay for us to not be a superstar right now. It's okay to really take your time during graduate school and take the time to learn what you need to learn. And I think we're all high achievers, and that's why we're tackling a PhD program. And that's where it really comes into play, where when you're really trying to push for a lot and trying to learn very quickly, it can really take a toll on the mental capacity and your capability to learn. And I found it very much more relaxing when you can kind of take a step back and be able to take the time to learn what you need to learn. And then to a similar vein to that, I think that graduate school really shouldn't be a lonesome process. It should be a very collaborative one. A lot of times, I think from myself, my personal experience, and also from a lot of my friends and colleagues, they've all spoken to the fact that you might a lot of times find yourself in a project or a experiment that only you are the expert on, and you are fully aware of all the different failure modes of your experiment, and you can get very easily frustrated. And I find that it's best when it's done in a very collaborative process. So I had this opportunity to work on a very unique project that was what became the science paper that we published. But what was really unique about it was that we worked in a very collaborative team. I think a lot of times in an academic environment, each individual might be responsible for their own project, and they are the lead author. But what was really unique about this project was that we, as a team of six or seven, really worked extensively on each part of the paper together. And so everyone that's on that list of authors really is a equally contributing author that's made the whole thing possible. And that experience to me was extremely rewarding and very fun. A lot of times you find yourself struggling with an experiment. You can go to someone that's keenly aware of the same problems, and they can give you advice, and maybe sometimes just to tell you to go home sometimes and just say, wait till tomorrow and come back and we'll figure this out together. And that's why I really feel like moving towards a research environment that is very collaborative. And of course, we were all aware of that publications have a list of different authors, and first to the last author. And I think it's tough for that reason, but I think if you can have the opportunity to find a collaborative process, even if it's just with a co-author, someone that shares in your project, I think that is hugely beneficial to not only research output, but also to kind of the enjoyment of that research process. And then also very similarly, finding the right people and the right work that is for you. I think the people and the work, to me, are the two defining characteristics of anything we do. And this is, I think this piece right here is really what I find the most common in graduate school and also in a startup environment. I think one, you need to find the work that really motivates you, that gets you going, gets you out of bed in the morning, and then that you can continue to grind through kind of the multiple experiments that you have to run. And then finding the right people that are as excited as you are about the work that you're doing. And then also for me, it's finding highly intellectual people that are at the same time very friendly to work with. Being able to talk to them very easily and to say, hey, I messed up here. What do I do next? And they're able to give you advice objectively and help you as a team together to achieve that. And so I think that applies both to research in an academic environment and also in a startup environment. And then lastly, I think this is something that I'm still working on. And so I think this is really a very key point to the work that I'm doing now, is kind of balancing importance and urgency of your work. So this is kind of taken from this idea of the Eisenhower box, where you have things that are very, very important and very urgent, things that are very important and not urgent, things that are not important but very urgent, and then also things that are not important and not urgent. And so kind of categorizing all the things that you're trying to juggle, whether in research or also in a startup environment, that I think is a really good way to balance all the things that you're trying to do and help you make better decisions on what is the best thing for me to do right now to spend my time and energy on, since time really ends up being our most important resource in both cases, right? Where in graduate school, you're trying to finish over your 5 or 6 year program, and then they start environments, you're trying to make the best decisions quickly, but also an informed decision. And so balancing that importance and urgency really is key to that. And so that's kind of just sharing some of my thoughts on navigating graduate school and how that kind of translates out towards developing a company from your work. And so we'll kind of shift gears now and talk about what we're doing at FluidForm and how we're sort of thinking about bioprinting. So I think as a company, I think our thoughts on bioprinting can be boiled down to two key problems that we think really needs to be solved for us to progress. First, I think in the field, a lot of people are working with a unique material that really has desired properties for a certain function. And so this could really range from different materials, from electroactive materials that needs to be integrated with excitable tissues, or it could be some sort of tunable stiffness material that changes over time, that allows you to then control the microenvironment of cells that you want to study. However, most of these materials, I think, turn out to be very difficult to print and assemble in 3 dimensions. And as a result, a second problem emerges. So it's not possible then to fabricate many of the desired geometries using these materials. And so what we've done at FluidForm is now made it possible to print these materials in any geometry. And so I like to show this image here, and I promise I will talk about what we've done to make this all possible. And I like to show this print here to kind of highlight the capabilities of our technology. And this image here is showing a scaled down 3D bioprinted model of the human heart that we printed. And so this model is derived from an MRI file printed using type 1 collagen as our material of choice. And so some of you might be asking, what exactly is 3D bioprinting and where did it all come from? How come at every single conference now I'm starting to see bioprinting, and what is the kind of the attraction to it? So shown here are some, I think, fairly recognizable images now at this point of 3D printed constructs. On the left is a vase printed out of a thermoplastic material, and on the right is a bunch of printed metal components that are laid out. So all of these parts are produced using 3D printing processes that have been developed around the intrinsic properties of the materials themselves. So on the left, you have this kind of hot glue gun method where you're melting a thermoplastic filament and pushing it out of a nozzle onto a platform, and then very rapidly solidifying and cooling onto the build platform so you can build the object layer by layer. On the right, you have metal powders that are inside a container that you then hit it with a very strong laser, which then sinters the metal powders together to then create complex 3D printed parts. And so for a really long time now, a lot of people in the field of biomedical engineering have asked this question: could we borrow these same 3D printing technologies that allow us to print plastics and metals and print biomaterials that together create a complex 3D printed part? And I think the answer is both a yes and a no. We can borrow these same technologies, same hardware and software, but the materials a lot of times end up being very difficult to print. And these materials, these biological materials, are a lot of times very soft and also very difficult to assemble. So we're talking about a lot of the very familiar biomaterials that are very commonly used in tissue engineering, such as cells, collagens, fibrinogen, matrigel, and then some of the PEG celluloses. So a benefit that I think plastic and metal printers take for granted is that the materials they print are a lot of times far harder than the soft water-laden hydrogels that make up our body. So plastics and metals can be heated and cooled very quickly to form them into very stable shapes. However, with these protein hydrogels that are listed on the left here that make up the majority of your body, the same cannot be said. So an analogy that we like to make is a watch assembling problem. So if I were to give you a disassembled Swiss watch, you can probably rebuild this. And this is because while each of these individual parts are small, they are rigid. So with some time, some knowledge, and some practice, you can most likely learn to put everything back together the way they were, despite the very complex arrangement of parts. But if you imagine these parts are now soft, they become actually very difficult to assemble. And if they're intertangled with each other as well, the level of complexity very drastically increases. And you might end up with the clock scene here in this very famous Salvador Dali painting where everything droops and slops all over the place across different surfaces. And this is actually exactly what we see in a print when we try to print a collagen 1 solution into a dish. So here is a very common 35 millimeter dish. We're extruding a collagen 1 solution onto the build platform. And what we had intended to print here was a very simple, just a rectangular slab of a couple millimeters tall in thickness. And so you'll find that the corners aren't sharp as a rectangular slab should be. Everything kind of coalesces and you can't really distinguish individual filaments that are being printed, which is an indicator that you have very poor resolution and print fidelity of these prints. And I should mention that each of these materials listed to the left here really have come with their own unique printing challenges that are very closely related to this rheological property, their gelation mechanisms, and also their gelation kinetics that needs to then be figured out. And so this is really the first core problem that makes bioprinting so difficult. And what we've done really as a field is to look at each of these materials individually and assess what makes them difficult to print, as I mentioned before, and then modify the materials for printability to make them printable. So you can imagine you start out with something that is very soft and then very difficult to print. What we've done as a field is to take these materials and modify the material chemistries individually to tune the gelation mechanism and kinetics so that they can be rapidly deposited and also rapidly gelled onto a surface, so then you can then stack these gelled solidified filaments onto one another so you can build a three-dimensional structure. You can modify its material composition to tune its rheological properties. So add in additives that allow the material to stand up in air, and then so therefore you can then stack filaments on top of each other, or you can print additional scaffolding materials for structural support. And a lot of times there are some drawbacks to these approaches. So for example, when you modify material chemistry to tune their gelation mechanism and kinetics, what might happen is that you end up reducing the cellular binding domains and then also changing the hydrogel stiffnesses to be either too soft or too stiff, which might not be what you want when you want to ultimately have cells interact with these constructs. When you try to modify its material composition, you may be adding in different components to be rheological modifiers, which can then ultimately lower the concentrations of everything that's mixed together. And then when you have these very stiff structural supports, you need to figure out a way to then deal with these structural supports. Do you leave them in when you have these constructs that need to be implanted into animals? Do these will end up affect the ultimate performance of your construct? And so this kind of leads us to the second problem. Because we need to so finely tune each material so that they become printable, we usually don't really have quite the perfect printable material. So we're usually in a between point where materials are somewhat printable, but they need to be printed in simpler geometries designed around the challenges of printing them. So you have these desired geometries that cannot be printed with your materials because the materials are soft. So a lot of times these simpler geometries might just be simpler shapes than what we had hoped to print, and do not really lend directly to some of our research goals. And so to quickly summarize these two core problems that we see and that we think we see in bioprinting is, one, you have unique materials that are very difficult to print, and two, you have desired geometries that cannot be printed using these materials. And so you might have noticed that we haven't even begun to consider the functional properties of the constructs that we're printing. So as engineers, a lot of times our design process starts with kind of outlining the functional criteria for the thing we're trying to create, then picking the materials and geometries that best create that thing. However, currently in bioprinting, our process is reversed. We're spending a lot of time making a material printable, finding a geometry that is printable with that material, and then assessing the function of what's created using this very specific material and geometry combination. And so and frankly, I think at FluidForm, we want to change that thought process. And before we dive into how we're doing that, I want to kind of invite you onto this brief journey, a thought experiment, if you will, that I think is pretty fun. So if you were to imagine all these problems that we discussed didn't exist, and you can print anything you might possibly want to using so what would you print? So what would you print using the right materials, the desired geometries, and explore the printed function of this material and geometry combination? So you might think about printing the vasculature system of a full organ. So starting from very small capillary scale vessels up to arterial scales that change and bend and bifurcate in many directions, and then have these vasculature networks expand and fill the entire shape of a full organ, such as the kidney seen here. The bone is another example that I think is really cool as a problem where it has different scales of architectures and patterns. So in this image shown here, you have the trabecular bone, which forms the fine mesh structure that's found in the center, and then the cortical bone that kind of lines the outside of this trabecular bone. And so being able to create these different scales of architectures that have individually their individual functions is really interesting. So what about a composite of multiple tissue types? So you might imagine in this image here of the hand, where you might imagine having different tissue types from skeletal muscle that needs to interact with tendons and tendons that then, and then you also have the ligaments that interact with the bones, and all this together then work together in concert to allow your hand to function as it is. Also, there could be ways to print devices that need to replace native tissue functions, such as the heart valve shown here. These devices need to function on their own, but they also need to interface with the soft tissue around them. So figuring out the device itself and also the interface is a unique challenge that could now be very easily printable. And you might also start to design and really dream up new geometries that don't exist in the body. So in this image to the bottom here, this is a kind of biomimetic looking design, but it certainly does not exist in our body today. And so these are really kind of the number of scientific questions that were previously difficult to study without 3D printing. But these are exactly the questions that we are interested in studying, and the ones that we kind of hope the world might develop the answers for using our technology. So we think we've developed this technology that now allows us to print at this level of complexity and to actually start to ask these scientific questions. And so this technology is what allows us to print with soft materials in any geometry for function. And so we're talking about the title of this talk, which is Freeform Reversible Embedding of Suspended Hydrogels, or FRESH 3D Bioprinting. FRESH is really a new kind of 3D printing known as embedded 3D printing. So in embedded 3D printing, we're no longer printing 3D printing in air, but we're printing inside of a support material that kind of cushions our soft ink while it gels in place. And this exact same layer by layer printing method occurs just like it did in plastic printing, only this time it happens inside of our support material. So shown here in this image, we have a syringe containing a soft material bioink that's dyed blue. And then we're pushing this through an extrusion needle. And then we have our printed object shown here, the letters FluidForm. And then all of this is done inside the support material that I'm talking about. And what is unique about this support material is that it is a yield stress material, meaning it will flow like a liquid with the application of a force. And with the removal of that force, it will behave like a solid. So this is analogous to, toothpaste also has the same yield stress behavior. So while it's in your toothpaste tube, it will remain as a solid. When you apply a force to it, you're able to squeeze it out and deposit it onto the bristles of your toothbrush. And then when you turn the toothbrush around, it won't fall off your bristles because of that yield stress behavior. And this yield stress behavior in our support material is made possible because it's made of these gelatin microparticles that are packed very closely together to then allow it to flow past one another while your needle is moving past these microparticles. So I think this process is very simple and intuitive when you're able to watch it happen. So here's a video showing this process happening. So we have in the syringe, we have a blue ink, an alginate ink, which will gel in the presence of calcium ions. So we very simply added calcium ions into the support material, which is in yellow inside this container. So if you watch this happen here, you'll find that we're moving the needle very quickly, depositing the letters FluidForm again. And then the blue alginate bioink is being deposited by layer by layer. And then once the print is complete, we are able to then melt away the support material made of gelatin microparticles, which will melt at 37 degrees Celsius to then remove the fully cross-linked alginate print. So the alginate, again, will gel inside the support material in the presence of these calcium ions. And this is what allows us to print for function and print with any gelling material and also print in any geometry. And so for these next several slides, I will show you a lot of the images and data from our work in science to highlight what is possible to be printed. And first, to kind of go over the development timeline of Fresh. In 2015, we initially described the process in the paper Science Advances and a couple other papers, and this is where Homan had utilized Fresh for his work for printing elastomers. And then we then followed up our work with significant improvements to Fresh in 2019 and published this in the journal Science. And then this is where then FluidForm was able to obtain an exclusive license to then from CMU of this patent to then allow us to leverage this technology for our work. And so here I want to show you this first example of what is possible using Fresh. And this is the idea of printing multiple materials with orthogonal gelation mechanism. So when I say orthogonal, I'm talking about very distinct gelation mechanism. So you may imagine you have 4 different materials you want to print. In this case, just an example print is made of collagen, methacrylated hyaluronic acid, fibrinogen, and alginate, and each of them have very different gelation mechanisms, with collagen being pH-driven, methacrylated HA being light-based, fibrinogen being enzymatic when cross-linked with thrombin, and then alginate being ionic, as we previously talked about. It will gel in the presence of calcium ions. So it's possible to do all of this inside Fresh because if you remember that we have these gelatin microparticles, and surrounding the particles, we actually have this interchangeable fluid phase. So this fluid phase can really be changed to anything you want. So it could be a pH neutral buffer that allows collagen to gel. It could be, we could very simply slap on a UV light, and then we can incorporate thrombin and calcium. And so in this image to the bottom right, we actually were able to print a 4 material print inside a single print with the letter CMU with the colors corresponding to the materials that were listed on the top left. And so this is not possible previously to be printed, to be all done together in air. It's a very challenging print, but it's actually very simple to do all inside a support material. And so in terms of the basic building block features that we can print using Fresh, we took collagen type 1 as an example material to explore this because collagen is the most abundant protein that you will find in the human body. And it really has this very large footprint in biomedical research to kind of where there's a big drive to understand its role in biology and disease and leverage this understanding for therapeutics. So here we're printing collagen type 1 filaments, where we're able to demonstrate that we're able to very easily control the diameter of these filaments from 200, 150, 80 down to 20 microns in diameter. And we can very easily pack these filaments laterally and stack them vertically, control their spacings of these filaments, and then leverage all of these capabilities to then build up towards a 3D assembly of filaments that will then turn into macro scale. All of these printing capabilities, we kind of wanted to see how well all of this holds up inside a large print over multiple lane scales. So we took the heart as a proof of concept model for this project, given that Adam's lab is particularly interested in cardiac tissue engineering. So we started with an STL model derived from an MRI file. So here is a MRI file of a human heart scaled down to 55 millimeters in its longest length, which is about kind of the size of a really big strawberry. And we printed this heart model directly out of collagen. And you can see that we created a lot of the features that you see in the file to the left here. And when we take the cross section of this print, we find that we're able to recreate a lot of the fine features that are found inside the file. So here we're recreating some of the trabeculate structure that have been found to be very important for contractions and also crucial in development of the heart. We're able to also very clearly demonstrate printing of these rectangular infill structures that we've chosen to fill in the walls of these prints. And so you should be able to appreciate the detail created in these prints from these micro CT images of the printed heart in the last slide. So we took the heart and put it onto a micro CT and scanned it. And you can find that we're able to create the right and left ventricle with the trabecular structures I talked about, as well as the 3D printed rectangular infills that we talked about. And also, we're able to recreate the pulmonary valve as well as the aortic valve, both complete with their leaflets. And also, what's important is that we have these chamber walls are able to be printed as a solid layer that is crucial in the heart for maintaining pressure and allowing your heart to not just explode. And so the heart, of course, was just a scaffold. It didn't have any particular function other than as a demonstration of printed capabilities using fresh. So we really set out to think about how to recreate function in some of the key components of the heart, with the first one being the valve. So we took a valve SCL model that we downloaded from NIH 3D Print Exchange. We printed it, and shown here in different views, you can find the valve being printed at high fidelity and also with the leaflets intact. When we handle it in air, in Andrew Hudson's hands, one of my co-authors, we were able to show that the valve leaflets are intact and patent, and they're able to open and close as the function of these leaflets are. And so we stuck this inside a pulsatile flow loop and showed that the valve leaflets are able to open and close as intended. Now, of course, this is very early studies, and this was only over a span of a couple minutes. But you can imagine that there's a lot of exciting work to be done moving forward to optimize the geometry and the print design file, and then also optimize that for function of the valve specifically. The next critical piece to this puzzle of engineering heart tissue is that the heart, just like any other tissue and organ in your body, really needs a very dense network of vasculature that supplies blood to its cells. So one of the key challenges in tissue engineering for a long time, and still is the ability to fabricate this vasculature network in 3 dimensions with control over the organization and geometry of these vessels. So we decided to take a computationally generative approach, because when you take MRI data like this, a lot of times you don't get the fine detail resolution that you need for the vessels, for the capillary and the arterioles and the arteries. So you might be able to resolve some of the larger arteries seen here. So we took a segment out from the MRI data shown here, which is the left ventricle, and we're able to populate that volume with a network of computationally generated lattice network. And we utilize this lattice network as our vasculature structure. So we took a piece of that and isolated that for our printed components. So shown here is a model, a see-through model of our printed vessels. You can find different vasculatures going from a couple millimeters in diameter down to a couple hundred microns. And then here is the actual print printed out of collagen. And in order to visualize perfusion of these vessels, we took this print and we optically cleared the print. And we flowed a very simple blood mimic, which is glycerol and water solution with dye through our vasculature network. And we find that each of the vessels are patent and are perfusible. And we have our print file was designed to then leak blood out, or not blood mimic out from the side of the ends of the construct. So you can see again here, we can perfuse all throughout from our larger vessels down to our finer, higher resolution vessels. And this is really exciting because when combined with additional data that I'm not showing here, we find that when constructs are implanted, fresh printed constructs are implanted into animals subcutaneously, the constructs actually encourage angiogenesis, which can be, when combined with these printed components, you can imagine that we can print larger vessels and allow the body itself to develop some of the capillary structures that are required to really feed a very dense tissue network. And then, of course, a very key functional component to the heart is its contractile activity that allows the heart to pump blood, which is orchestrated by a very highly organized heart cells, cardiomyocytes. So here we created a simple model of the left ventricle, which is really just a football cut in half. And this is responsible for pumping oxygenated blood throughout your body. So we printed this out of collagen and human embryonic derived cardiomyocytes. So shown here is the actual print itself. And when we take a look at the walls of this print, we actually printed this print out of 2 collagen layers, sandwiching a single internal region packed full of cells. These cells are, again, human embryonic derived cardiomyocytes and human embryonic stem cell derived cardiomyocytes. And we decided to print these cardiomyocytes at a very high concentration, which was 300 million cells per ml, which is an order of magnitude higher than I think what a lot of tissue engineering research uses. And the reason we chose this concentration was that we really wanted our cells to be very densely packed into our construct, and so that they can very easily interconnect and form very tight electrical and mechanical junctions that are necessary for their functions. So we find that the cells remain viable after 14 days and 21 days of culture. And what's really interesting is that we find that when we take a closer look, we find that the cardiomyocytes really begin to align along the circumference of the ventricle wall. And this, we have a lot of hypotheses about why this happens. But I think it's really interesting that this cardiomyocytes really begin to align, as you might find in the native heart. When we take a look at the function of these cardiomyocytes, we decided to utilize calcium imaging. So calcium is plays an important role in excitation contraction cycle of cardiomyocytes and also in your heart. And we decided to use calcium using a calcium indicator dye to visualize the contractions of these cardiomyocytes. So in this video shown here, you'll find that we have these nice calcium waveforms that propagate across the entire surface of these ventricles, which is really encouraging because it indicates that not only do we have cardiomyocytes that span the entire surface of these constructs, they are viable and they are very closely connected, that the calcium waves can propagate across the entire surface. And you can see similarly from the top view of this ventricle model that we have this nice uniform propagation of calcium waves. And we can also assess or quantify some of the metrics from these calcium waves. So we can take a point electrode and stimulate that at a point, which is the red asterisk seen here. And we can quantify over time and distance conduction velocity of these printed ventricles. And I won't go into too much detail here, but I think all this together is really exciting for us because this really represents the ability to not only print cells and materials all together in a single print, but also start to recreate function that we can actually utilize. So start to study these functions and think about how we can engineer our print differently to create different models for ventricles, and also utilize these models to study disease and healthy tissues for cardiac tissue engineering. And I think with that wraps up our journey through using the heart as a proof of concept model for fresh 3D bioprinting as a technology. But all of this really is what I think is just the tip of the iceberg for what fresh will allow us to do as a field. I think we were successful in starting to recreate some of the key functional components to the heart using fresh bioprinting. And I think that sets us on the path towards printing tissues with more complex organizations of material and geometry, with this very clear goal of proceeding towards clinically relevant tissue engineering. And I want to show you here some of the work that we've been doing at FluidForm. We've been thinking about how we can take our technology towards eventual long-term goal of organ scale transplantation. So starting with some of these very recognizable parts of our body. So shown on the top left here is a different file of the heart valve, where you can more easily appreciate the valve leaflets that are printed intact. So here, all of this is printed out of collagen type 1, and you can see that the valves remain intact and preserve as print fidelity, and you can open and close the leaflets as we've shown previously. On the top right here is a model of the ACL, which is, as many of you might know, is very common in injuries, and a lot of times ACL transplantations are necessary by taking transplants from other parts of your body, either tendons or muscle tissue as transplants. And it's possible to now be able to think about printing these ACL components life size, and also preserve its mechanical properties so that it's able to survive transplantation and be utilized as a transplant. We can also, there's also a huge need for transplantable trachea when during injury, or even in pediatric cases where trachea deformities are really difficult to address. And so here we have a trachea with the lumen that is open, and this is of course a pretty scaled down, but you can see that it's extensible, and also it can be manipulated by hand. And then an intervertebral disc is responsible for the loading in between your vertebrae. And these discs are really what cushions your different movements as you move your back and through your range of motion. And a lot of times in the case of a herniated disc in sports injury, or through just daily practice or daily use, there is fracture of these discs, and the central region that you see here leaks out of fluid that then causes bulging of the disc that then leads to severe pain if not addressed. And right now, the current state-of-the-art treatment for these is printed using metals or using other polymers that are extremely stiff and might not be able to be integrated into your body very easily. And so this is kind of what we at FluidForm are trying to do. And we've seen a lot of others in the field have also kind of recognized the importance of this technology and really taken on to adopt Fresh as a key piece to their research. So we've seen a lot of peer-reviewed publications as evidenced by this chart. And these are just kind of a few examples from several papers published in the last 2 years using Fresh. And you've seen the number of Fresh publications grow by year. And so FluidForm as a company has really taken on this mission where we're really thinking about expanding the reach of Fresh, because we truly believe it can have significant impact on biomedical research. So our support material is commercially known as Life Support, which is really very simply a sterile powder that you can rehydrate into support material that you previously saw, and you can go from sterile powder to printing within 15 minutes. And what I really want to emphasize here is that Fresh is really very simple to adopt and slot into existing bioprinting research. As I mentioned throughout this talk, Fresh now makes most biomaterials printable. You can really think about any design, any geometry that you might want to print. This is usable with any printer and also with any software out there. And so this is what makes it so versatile and so easily to use. And so lastly, I just kind of want to leave my email address here. If you have any further questions about bioprinting or Fresh printing related, I'd be happy to chat. These are some of our collaborators that we're working with to really expand the use of Fresh. And then also, if you're listening to this talk, feel free to ask for a sample and feel free to ask for tips to get started. And I'd be happy to kind of help think about how to integrate bioprinting into your research, what to think about when designing a print, and really how to set up your first print, making sure all that turns out well, and really get you onto thinking about your bioprinting research as you might want it to be. So with that, I'd like to thank everyone for joining us today. I'd be happy to answer any questions that you might have.
Well, thank you very much, Andrew, for a very nice presentation that you had today. Pretty impressive work, very inspiring for all the graduate students and professors, and then also the members of basically industry. So before we get into the question and answer Q&A part, first of all, make sure that you ask your questions in the question box. We have plenty of questions, so please vote to make sure that, I mean, those questions who have the most vote will come first. And then one other thing that I wanted to mention is that next week's speaker is Dr. Massimiliano Paganelli, the founder of Morphocell Technologies. Will give an interesting talk on the platform that they developed for stem cell-derived encapsulated liver tissues. I also would like to acknowledge the support we received from Transmed Tech. So Transmed Tech Institute is goal is to support the development of innovative medical technologies and train the next generation of professionals and make innovation in life sciences and engineering a source of wealth for society. I would also like to acknowledge the support that we received from University of Victoria, in particular University of Victoria's research partnerships and knowledge mobilization team, which facilitates the development of research partnerships, commercialization of university-developed technologies, and also support for new startups. So with that, wow, we are receiving many comments and questions. So let me just start with a few questions. Okay, so Chris Flores from UVic. So Chris's question is, so first of all, he thanks you, Andrew, for sharing this very interesting approach, bioprinting soft materials within the support material. His question is about the top two challenges associated with this approach. I wanted to know your comments about these challenges.
Yeah, I think that's, I think typically the top two challenges for me, I think one is kind of integrating every part of the bioprinting workflow, right? I think as a lot of biologists, we might not be as familiar as we need to be, or as familiar with kind of like printers, what sort of printers to use, what sort of software, and then how that all plays together to work into our projects and materials that we want to print. I think that's kind of the challenge of like integrating, and there's no really existing protocol workflow out there that helps you develop that whole process. And I think that's one of the first challenges that's unique for each individual labs out there. And so each lab will have a different project. They will have a different application for their project, and also they'll have a different printer. And I think what Fresh does is that it makes kind of all those things ubiquitous. So that's a first challenge. A second challenge, I think, is a fun one. I think it's figuring out what you want to print. I think it's a really interesting one, because now we can actually think about any sort of possibilities, any sort of combinations of materials, of cells, of geometries. And it's actually kind of mind-boggling how difficult that problem is, because we're now not limited to anything else. We can, like, I have a hard time a lot of times to think about do we just want to copy what nature has to offer us, which is really optimized through evolution to be very, very, very efficient and effective, or can we as engineers think about ways to design our own functions into what we print? Use simpler materials, or maybe we use more complex structures, more complex functions. Can we engineer much more different functions into a single component? And so that is actually a really fun one, but a really challenging one that I think will take time, and it will take the creativity of kind of our entire field and interdisciplinary research to kind of put everything together. But this is what we are now capable of doing. I think it's really cool.
Wonderful. So the second question is mostly about the inks, the types of inks that are printable, and in particular, Ketolytic is asking about whether this method is compatible with chitosan. Have you ever printed chitosan, and if so, did you find advantages related?
Yeah,
to
so
its
we
positive
haven't.
charge?
I haven't personally printed with chitosan, but chitosan, of course, is very attractive for different uses in tissue engineering. I think what you can think about in terms of the charge is, can you pair it with a negative charge material that allows you to assemble in a very very unique way, right? And so I wouldn't be able to tell you off the top of my head how to do that, but I think it's certainly compatible. And what you really only need to think about is, can you put it into a syringe? Can you deposit it into a solution? In what sort of buffered solution, or what sort of solution do you need to print it into, allow it to gel, right? So you can certainly print it out of a syringe needle, and then it's then up to you to then decide how you want to use it. So.
Right. Awesome. Well, I mean, before I get into other question, I have my personal question that I wanted to ask you. What are your thoughts on the future of 3D bioprinting, and particularly in the context of translational medicine? Where do you think the field is moving towards?
For a lot of years now, we've heard about 3D bioprinting, right? And how much it can do. We can print now human hearts and organs, and I think we're still a little bit ways away from that. We're certainly not ready to, there's a lot of biology that we don't understand. We are trying to learn how to leverage cells to do what we want them to do, but I think it's, to a lot of people on this call, it's very difficult to do. There is a challenge of generating enough cells so that we can actually print something that is large enough, right? That's one of the challenges. And then printing, and then supplying cells with vasculature network to help them survive and help them continue to do what they do. Those are kind of the very unique challenges that we need to solve first. So I would say we're still, 10, 20 years away from being able to get an organ that we can start to study and start to investigate even in animals. And so certainly much farther when trying to think about implanting into humans, since there's a lot that we don't understand. But I think in terms of translational research that we can do, and taking this out into just being able to think about how to develop our technology for a clinical setting, I think that requires is a very interdisciplinary approach, right? So taking not just our biologists, not just our engineers, but also some of the designers that can help us think about ways to design this effectively, and then also taking some people have thought about using a machine learning control so that we can very quickly adapt to defects in our prints. And then also we can use an AI generated approach to then to think about best ways to optimize our designs, best way to utilize our materials, and predict through just some of the work that's been done in finite element analysis to predict what cells will do to your structures, how that then ends up maturing, and then what happens when they mature. Does your structure ultimately buckle or deform, or do they remain in place? And so combining a lot of different fields together is, I think, what will allow us to move towards a more translational setting.
Wonderful. I agree. So there's another question from Kosar who. And it's about the. The question is, how can you handle the temperature sensitivity of collagen while printing? How do you make sure that it is not denatured?
Yeah, that's a great question. So collagen type 1 is typically soluble in an acidic condition, and so we keep the collagen in its acidic condition, and it's pretty stable inside a syringe that way. So it's kind of this viscous fluid that is kept inside the syringe. And then we print it into the support material, which is filled with a buffer. And so the buffer very quickly neutralizes that acidic solution of collagen. And once the collagen is neutralized, it will then very quickly gel into, or polymerize and self-assemble into a solid material, right? So we have this solution to gelation, sol-gel transition that is what we leverage in our process. So keep going from a solution to then a solidified structure. So that keeps us from having to deal with temperature sensitivity of the collagen. But that's certainly something that you can additionally leverage to then further polymerize the collagen once it's printed or assembled.
Great. So the next question is about the surface topography or surface roughness of the bioprinted blood vessels. So the question is whether you have you see any surface roughness, and then whether you measure the roughness, and then if there is a roughness, do you think it will have a significant effect on angiogenesis?
Yeah, that's a really, I think, insightful question. So on our filaments, we have, so say you take an individual collagen filament, because it's printed into the support material made of gelatin microparticles, the particles kind of impart this kind of crater-like topology onto the filaments. And we haven't studied extensively on how that topology affects cells, but there's certainly been a lot of studies on how different surface topologies, how cells will interact with those surface topologies. And so I think that's something that's really interesting to study. That's not something that we've done, but you can probably extrapolate some of the work that's been done out there on creating different scaffolds, different topological cues that help cells, that help direct cell function or cell migration. In terms of angiogenesis, what is interesting is that what we found was that, again, I think I mentioned this very briefly in the talk, is that the porosity that these microparticles coincidentally impart into these constructs was what allows us to control angiogenesis inside the construct. So we took a very simply printed disc of material implanted under the skin of a mouse and found that it performed much better than in terms of vasculogenesis or angiogenesis into the construct in comparison to a casted collagen disc. And so that is really interesting for us to leverage, because we can use the body's natural angiogenic system to then create capillary scale structures, which are very difficult to print and very difficult to manufacture or engineer in certain ways.
So Homan has a question.
Homan has a question.
Michael, to ask a question as an organizer.
Thank you so much, Andrew, for the presentation. I just want to switch the gear now, going to your entrepreneurship journey. Maybe like the audience, like me, they are also puzzled. You are a great academics. You publish in science. Why you wanted to be an entrepreneur, and also how do you like it? How was it so far?
So I found it a lot of fun so far. So the company was founded while I was still in grad school. So I was probably a year away from finishing before the company was founded. I joined so probably a year after the company has been founded. So we have 4 of the co-founders. And what I think what's really different about a startup environment and an academic environment is that it's really, really fast moving. A lot of times in academic research, you might kind of feel like there's this infinite timeline. Things just kind of go on forever. But there's certainly timelines to being able to submit grants and submit your publications. But I think from the viewpoint of a student, you feel like you're constantly working and there's no end goal in sight. But in a startup environment, that's kind of really very opposite to that. It's very, very fast moving. You need to very quickly decide what you want to do next. As one of my mentors kind of told me, is that generally what you kind of look for in a successful entrepreneur is you want to make, on average, more better decisions than bad decisions. And so that's very, very wide open, but I think that makes a lot of sense to me, because you're never going to have, that's one of the problems I ran into, is that you're never going to have all the information that you want, right? So that's one of the biggest challenges for me moving from an academic environment to a startup environment, is that in research during my PhD, I've always wanted to know everything, understand everything, be able to find out answers to every problem of my experiments that went wrong, and be able to figure out everything. But in a startup environment, because it moves so fast, you don't have the time to do all of that. You don't have the time to figure out every single detail about what problems you're having. And so you need to make very fast decisions very quickly. And so that was very difficult for me, just transitioning. And that's something that I'm still learning to do, because I'm kind of academic background, that's what I tend to do. But it's been a lot of fun, just because you get to do so much. You get to be exposed to not just the research part of things. You get to think about the business itself, right? How do you not just you need to do the research well. You need to have this platform technology, but you need to also think about ways to how does it fit into a market? How does it fit into a business plan? And then not just for your customers, but also for the investors that you're looking for. They're looking for potentially different stories that you're telling them about your technology and how that might impact their future work. So your different stakeholders is what you need to consider beyond the research itself.
Wonderful, wonderful. I really like your statement, and entrepreneurs need to make many good decisions, and then they. I mean, it's not very good to make bad decisions, actually. So you don't have the option of making many bad decisions. Whereas when you are in academia, we may sometimes make bad decisions. I personally. But this is very very interesting, actually, quote from you. It's going to go on my LinkedIn and Twitter, referring to. So the other question that I have is, again, most of the questions are about the technology. For example, Vahid is asking about how the shear stress and cell viability can affect the cell viability and cell function, and can you comment on the effect of resolution when you want to print like very fine fibers? The question is whether that will have any impact on the cellular viability, and if so, how did you overcome this issue?
Yeah, so that's a question we get a lot in terms of cell viability when pushing it out through a very tiny needle of a tiny nozzle diameter. So typically, a lot of our prints are printed out of an 80 micron inner diameter needle. Cells are around 20 to 40 microns in diameter, depending on the type of cells that you have. And there certainly is concern for cell viability, but what we found in a lot of our research is that it actually does not affect the cell viability too much, pushing it out of a needle. And so we kind of showed that in the ventricle print I talked about, where we're printing out of two syringes, one printing a collagen ink, and right next to it, we're printing a cell ink. So the really high concentration cell cardiomyocyte ink. And we did a very rough cell viability study looking at what happens after we deposit it, and we found actually really, really good viability, so 99%. And that was really, well, actually 97%, I believe. But that was really encouraging because we don't lose a lot of cells that are so precious to us. So I think that's not something that we've seen. In terms of the resolution of these really fine filaments, I think this is kind of related to the topology question. Is what do cells do when they see these really fine filaments? And that, again, is another research question that we can study. There has been a study out of University of Florida, Thomas Angelini's group, where they looked at individual filaments and how cells pull and bend on these filaments of collagen filaments in a similar approach. And so those are kind of the cellular mechanics studies that are really interesting to me as well, right? Can the cells crawl along these filaments? Can they bend these? And then how do you leverage these actions of the cells to then create a final structure that you want or that you want to study? Mohsen, you're muted, I think.
Oh, thank you. Thank you very much. I thought I'd unmuted my microphone. So let me just ask you another question about the importance of mentorship. You talked about benefiting from excellent mentors in your career. Can you elaborate a little bit more on how important it is for graduate students, early career researchers to communicate with their mentors? How do they pick their mentors? How do they interact with them at different levels and engage them in their research?
Yeah, I think that's a really difficult question. But from my own experience, I've had really great mentors. And I think the reason for that is because the mentorship style kind of fit me. And then I've also taken to think about how do I use that? How do I get the most out of that mentorship, right? So usually I like to kind of do my own thing. And so I like a very hands-off mentor who will give me very broad feedback. And so, but that's just me. I think what you really need to figure out is what kind of person you are in terms of a mentorship-mentee relationship. And that's not easy to figure out, because a lot of times when you start a graduate program, you might think that you want someone that's hands-off, but what ends up happening, you might find that you really do need a lot of guidance on directions you need to take next, or even just things outside of research, things that will help guide you to do better research, right? So some of your mentality. And so different mentors do different things, I believe. You have research mentors and you have kind of these mentality mentors who can help you just shape who you are. And I think it's natural to struggle with that a little bit, because coming out of college, there's no experience that you have that really trains you for that, right? And so that's kind of why my point of just take your time and figure out some of these things. That's what graduate school allows you to do, and mentorship is only a very small part of that. That's not easy to do. And then I think this is where the mentee or the early career graduate students then need to take the initiative and say, hey, this is not where I'm very good at. Even just say, I don't know what type of mentorship style I need. Can you help me with this? And I think most people in academic environment, they're very helpful. It could be your fellow graduate students. It could be postdocs. I think most labs, most research settings aren't competitive, right? You're trying to help each other succeed. And so this is where a very safe environment for you to just ask those questions and say, I don't know how to do this. Can you teach me, start me on the path to learning about this? And I think that's the most important part for the student, is to just ask. And for me, it's not easy to do. It takes a lot of courage to just say, yeah, I don't know how to do this. I'm not very good at this. And that's certainly something I encourage people to do, something that I'm still working on as well. But something that I think is really important.
Great advice. Great advice. This is very useful for graduate students who might be on this, listening to your talk. I agree that it needs a lot of courage to ask someone for help, but it's okay, especially in this field, which is a multidisciplinary field, bioengineering, and you are not expert in everything. So it's okay if you seek for help from others. and it's okay if you share authorship and the credit with others. It's all okay, and then and that's great to hear it from Andrew, actually, who has been very successful, and then who developed this groundbreaking technology. That's great to hear. Again, I mean, these are great pieces of advice for graduate students and then early career researchers to know that collaboration is very important. It's extremely important, and it helps you to grow faster than when you work in your own silo, and then you think that you can solve all the problems by yourself.
Yeah, definitely. I think I like that silo part. Like, you don't want to just be stuck in your own path, and you don't get exposure to different perspectives, and you aren't able to improve as fast as you want.
Exactly. So we have time. I don't know how much time do you have, Andrew. I know you're very busy these days, so we can ask a couple of more questions. We have so many questions, but I think in the interest of time, I'll go with one or two more questions if everyone agrees, and then we will conclude this session. So the next question is about the size of the gelatin microparticles, and whether the size has any effect on the resolution and the printing process.
Yeah, I think that will depend on your application. A lot of times you're printing a structure that doesn't need such fine resolution. So the size of the gelatin microparticles are around 30 to 50 microns. But I think resolution is, again, just a word for how do you want to use that resolution, right? So do you care about cells interacting with these very small features inside your print? But it certainly does not affect the assembly of the prints. So you can see that a lot of the larger constructs, they turn out visually looking very well. And then so controlling that size is also fairly easy to do. And so it's a fairly open-ended question, I think, that resolution really will depend on what you choose to do with it. So it's tough for me to answer that. But I would say overall, it's not something that we focus on a whole lot, just because it can be controlled, and it's something that will vary depending on what you choose to do with it.
Yeah, great. So the last question, if everyone agrees, is that about the highly perfusible organs. So the question is from Sylvia, and she asks about your experience with highly perfusible organs with very intricate tubing structures, such as kidney. What are your comments on these?
I think the challenge with highly perfusible organs is that, like I mentioned before, it's hard to get a file or a printable file that captures all of those intricate vasculature structures, right? And so the challenge is, how do you design something that's then kind of replicate that function? Can you engineer it? Can you print it all directly, which might be unlikely? And then utilizing that for your research. Another challenge is the perfusion part, right? It's optimizing the flow of liquid through these different channels is very difficult. Our body does a very good job of optimizing so that you don't get pressure buildups, so that everything kind of flows smoothly through these organs. And then so that's actually something that is actually a complex engineering challenge that needs to be figured out. And so it could be taking the computational approach to figuring that out. It could be experimental approach where you build out different structures and then combining all of those to then create your final construct. Another piece to that is, I think, a lot of these tissues are very soft. Interfacing your tissues with your perfusion chamber is sometimes not trivial, right? You have these very rigid needles that need to be pushing fluid, and again, it's how do you manage that interface with your equipment on hand, and then perfusing that properly, correctly with the right flow rates, and then also dealing with the challenges of having cells in there, and am I flowing it too much? How do I seed cells into each of these fine detail structures? But those are, again, those are the fun things we do in research, right? So I think, yeah, those are just things to think about while you think about doing your work.
Awesome. So with that, maybe I think that was the last question. So I would like to thank you again, Andrew, for your great presentation. It was amazing to see how a big problem in the field of tissue engineering is being tackled by you guys. It's amazing to see that you were able to create such complex structures using a new technology. Printing heart is not easy. Everyone knows that, and then you guys managed to do a great job. So I would like to thank you for agreeing to give a presentation and our e-seminar series. So before we end this session, I would like to remind everyone again that our next speaker is Professor Massimiliano Paganelli. He is going to talk about stem cell-derived encapsulated liver tissues, preclinical development of allogenic cell therapy to treat liver failure. Dr. Paganelli is the founder and CEO of Morphocell Technologies Institute. I'm sure he has a lot of experience and experience in entrepreneurship that he can share with us. Please make sure that you attend his presentation next week. If you have any questions or concerns about this e-seminar series, feel free to contact me and Dr. Sabuji, who are organizing this e-seminar series. You can reach us through our emails, or you can contact us via Twitter. You can follow us on Twitter. Also, please follow us. Follow this e-seminar series Twitter account to get more the most up-to-date information about the upcoming speakers. And then last but not least, I would like to acknowledge the support that we have received from our sponsors, TransMedTech Institute, and then also University of Victoria, University in particular, the Research and Knowledge Mobilization Unit. So with that, I would like to also thank all the participants for attending this meeting, and then wish you a great day, and see you next week. Thank you, everyone.
Thank you so much, Andrew. Thank you, Mohsen.
Thank you.
Bye-bye.